Hello everyone. Today we will know about the nucleus. So, this is a cell, and inside a cell, a main component or structure is present, and this is called as the nucleus. So, the nucleus is, we can say that it is a main organelle of the cell. So, this nucleus it governs all the activity inside the cell. So that's why the nucleus is also called as the director of the cell. Director of the cell. Why it is called as the director of the cell? Because it directs or governs the whole total cell. So that's why it is called as the director of the cell. So now the nucleus, it was discovered by Robert Brown in 1831. So in 1831, Robert Brown discovered the nucleus. So look at the structure. This is a nucleus. So the nucleus has four parts. Mainly it can be divided into four parts. The first part is called as the nuclear envelope. Nuclear envelope. The second part, we can say that it is the chromatin material. Chromatin material. The third part is called as the nucleolus. Nucleolus, which is the main component of the nucleus. And the fourth one is called as the nucleoplasm. Okay. These four structure or the nucleus can be divided into these four parts. One is the nuclear envelope, one is the chromatin material, third one is the nucleolus, and the fourth one is the nucleoplasm. So first come to the nuclear envelope. Nuclear envelope. What is called as the nuclear envelope? It is simply like the plasma membrane or the cell membrane. Look at the structure. This is the cell. So this is the border, or it is confined by a border like structure, and that is called as the plasma membrane or the cell membrane. So like that, the cell also surrounded by a membrane, and this membrane is called as the nuclear envelope. So it is just like in the cell, the plasma membrane is present. So like that in the nucleus, the plasma membrane of the nucleus is called as the nuclear envelope. Look at the structure. If I zoom the structure of the nucleus, it looks like this. That means this is the nucleus, and inside it a structure is present, and this is called as the nucleolus. We will study about that after some time. So first come to the nuclear envelope. Look at the structure. So this nuclear envelope, it is double membrane. Double membrane. But the cell membrane, the plasma membrane, is the single membrane. It is the single membrane structure. But the nuclear envelope is the double membrane structure. So it is the outermost double membrane structure, and look at this structure. This is called as the outer layer. Outer layer. And the inside one is called as the inner layer. Inner layer of nuclear envelope. So the space between outer membrane and the inner membrane is called as the perinuclear space. Perinuclear space, which is the space between inner layer and the outer layer. So, and before that, the nuclear envelope, it is also called as the karyotheca. Karyotheca. So now come to again. So this is the outer layer, and this one is the inner layer, and the space between that is called as the perinuclear space. So this layer is not continuous. So that means, like in the cell membrane, let's suppose this is a cell membrane, this is the cell, it is the cell membrane. It is a continuous. but this membrane is not continuous. That means there is some divided in that, in some area, and this divided part of the pore like structure is called the nuclear pore. The nuclear pore. And the nuclear pore, if we ask what is the exact size of the nuclear pore, it is nearly 500 angstrom. What is the angstrom? 1 angstrom is equal to 10 to the power minus 10 meter. Okay. So the nuclear pore is the 500 angstrom, that means 500 into 10 to the power minus 10, that means 5 into 10 to the power minus 8 meter. So that means this is a 500 angstrom, 500 into 10 to the power minus 10 equal to 5 to the power minus 8 meter. So this is the size of the nuclear pore. So what is the function of the nuclear pore? Look at the structure. Let's suppose this is a nuclear pore like structure. So that means through this nuclear pore, the RNA and the protein can pass to the cytoplasm, or it can pass from the cytoplasm to the nucleus. So this is the main function of the nuclear pore, that is for the transport of RNA and the protein. The DNA never passes the nucleus, because the nucleus should remain inside the the chromatin or the DNA should remain inside the nucleus. So that's why DNA never passes the nucleus. Only the RNA and the protein will passes to the cytoplasm, or the RNA and the protein from the cytoplasm, it can pass to the nucleus through nuclear pore, which is the size about the 500 angstrom. So this is the outer layer, this is the inner layer. Look at the structure exactly. So at the outer layer, a structure is present that is called as the endoplasmic reticulum. So that means, let's suppose this is a nucleus, this is the inner layer, let's suppose this is the outer layer, and from the outer layer the endoplasmic reticulum emerges. So from this outer layer, endoplasmic reticulum emerges like this. Look at this structure. So that means the inner layer is present inside, but the outer layer is continuous with the endoplasmic reticulum. That means the outer membrane is connected to the membrane of the endoplasmic reticulum. So it is formed by it forms the endoplasmic reticulum at the outer side. So this is all about the nuclear envelope. So if you are watching my video, please like and subscribe my video. So now come to chromatin material. So chromatin material. So what is the chromatin material? So that means this chromatin material are present inside the nucleus, and this chromatin material is main component of the nucleus, or this chromatin material, because it contains the gene or the DNA. So that means this structure regulates our entire the cell. So that's why they are called as the director of the nucleus or director of the cell. So now simply, if in the exam it will be asked that what is the chromatin material, so simply you can say that the thin thread like structure that is present inside our nucleus is called as the chromatin material. And always the chromatin material or the DNA, so they all are the negative. So negative means it is the acidic. So because the nuclear material or the chromatin material is acidic condition, so now it is stained by the basic dye. So always you should know that the chromatin material, it is stained by the basic dye, because the chromatin material is itself the acidic, so that it can be only stained by the basic dye. So now the chromatin material, it contains DNA plus RNA plus proteins. So now this protein, it can be the histone protein or it can be the non-histone protein. Okay. So now come to what are the exact structure of the chromatin material. So you already know the word chromatin, chromosome, chromatid and DNA. So what are the chromatin, what is the chromosome, what is the chromatid and what is the DNA. So actually these all structure are the same, but the word the chromatin, chromosome, chromatid in the DNA, it differs because it depends this structure is present in which stage of the cell division. So now you should know something extensive about it. So in the cell cycle, let's say this is the cell cycle, this is called as the M phase. So this is called as the gap 1 or G1 phase. So this is called as the S for synthesis phase, and this is the gap 2 or the G2 phase, and it is the G0. Okay. So now these gap 1, S and the gap 2, these are called as the interphase of the cell cycle, while this M phase is called as the division phase. Why it is called as the division phase? Because only in the M phase the chromosomes are divided. So that's why this is called as the division. And why it is called as the interphase? Because during the gap 1 synthesis or the S phase or the gap 2 phase, and in these phases the chromosome doesn't divide, it only prepares for the division. So that's why it is called as the interphase. So now what is the structure of the chromatin, chromosome, chromatid? Now we can say that. So when the chromosome is present inside the interphase, that means gap 1 S phase and the gap 2, and this structure now it is called as the chromatin. And this chromatin structure, it is very thin structure. Okay. So now when it enters to the M phase or the division phase, so now this chromatin becomes condensed. What is condense or condensation? What is the condensation? That means simply you can say that the chromatin thickens, and it thickens and it can form a broad structure, and that is called as the chromosome, and now it is called as the chromosome. So that means the structure of the chromatin and the chromosome are the same, but the chromatin is present in the interphase, and during the presence in the interphase, the chromatin is present in the thin, or you can say that it is present in the decondensation phases. That means it is become a very thin. So now after going to the division phase, now it condenses and it enlarges or the thickens and to form now the chromosome. So that means the structure, if it is present in the division phase, then it is called as the chromosome, and if it is present in the interphase, then it is called as the chromatin. Okay. So another word is the chromatid. So what is the chromatid? So now look at the structure at the group at the gap 1. The chromatin structure is like that. Okay. In the S phase or the synthesis phase, here the number of the DNA doubles. So that means here it is called as the, that's why it is called as the synthesis. That means here the synthesis of the DNA occurs. That means the number of the DNA doubles. So now here number of the DNA doubles, and now it is called as the chromatin structure, and here also it is called as the chromatin structure. But in the gap one, the chromatin or the DNA or the number of DNA, let's suppose it is 1, and now the number of DNA is 2. So what is the DNA, what is the chromatin? So first of all you should know something about the DNA. Look at this structure, this is the DNA structure. Okay, so actually the DNA structure, this is simply a thread like structure. So this thread like structure, it wraps around each other, and now it forms the chromatin structure. So this chromatin, this chromosome, it is simply the DNA. So once this DNA, it wraps around each other to form the chromosome and the chromatin. So in the one chromatin, let's suppose this is the one chromatin. In the one chromatin, one DNA structure is present. That means here this is the chromatin structure. Here one DNA wraps around each other, and now it forms like this structure, and it is called as the chromatin. Look at this structure. This is the DNA. This is the simply the thread like DNA structure. Now it interacts with the protein, and this protein is called as the histone protein. Look at this structure. This is the histone protein. So now the DNA with histone protein is present, and this structure is called as the nucleosome structure. Okay, so now the DNA with histone protein, it forms the nucleosome. Now it again wraps around each other like that. Here it wraps around each other, and now it forms the chromatin structure. Okay, or the chromosome structure. You can say what is the chromatin and the chromosome, but it depends whether it is present in the division phase or in the interphase. If it is present in the interphase, then it is called as the chromatin, and if it is present in the chromosome, if it is present in the division phase, then it is called as the chromosome. So now in the gap one, here the chromatin structure is like that. Okay, but in that case, for example, in the division phase, it looks like this. So what is the double structure? Why? Because in the synthesis phase, here another chromatin structure or another DNA is synthesized. So now these two chromatins are joined together. Now it forms like this. In the book, you know this structure, and now this is called as the chromatin. So now in the S phase, the chromatin is present. In the gap one, the chromatin is present, but in the gap one, the DNA number is 1. But in the S phase, the chromatin is present, but here the DNA number is 2, because your one DNA structure, it forms one chromatin, and here another, it forms another chromatin. Okay, so these two, this is the 1 and this is the 2, or here 1, this is the 2 structure, and each one is called as the chromatid. This is the one chromatid, this is another chromatid. That means in one chromatin or the one chromosome, 2 chromatids are present. Here one chromatid, here another chromatid, and each chromatid are called as the sister chromatids. Sister chromatid. Okay, so now this is the one chromatid, this is the another chromatid, and they are called as the sister chromatid. So if we look like this structure, at the center one, it is called as the centromere. Centromere. Okay, so now in the centromere of the each chromatid, there is a one structure is present, and this is called as the kinetochore. Okay, here also one kinetochore is present. So what is the function of the kinetochore that is present in the centromere? It helps in the binding of the two chromatids, because this kinetochore that is present in the centromere that helps in the binding of the chromosomes, and now after this it forms like this structure. So that means the two kinetochore that is present and they helps in the binding of the chromosome. So now this is called the chromosome and this is called the centromere. So now it contains two kinetochore, that is very much important, kinetochore. So that means two kinetochore present in the single chromosome structure, because in each chromatid one kinetochore is present. Okay, so this is the main structure of the chromatid, chromosome, chromatin and the DNA structure. So now if we look at the chromatin structure, the chromosome structure, let's suppose this is the chromosome structure. This is the one sister chromatid, or it is called the chromatid, one chromatid. This is another chromatid, or simply they are called as the sister chromatid are present, and here one kinetochore is present, here also one kinetochore is present. That means here the two kinetochore is present, and this kinetochore that helps in the binding of the two chromatins. Okay, so now look at the structure of the chromosome, and here one constriction is present, or this is called the primary constriction, and this primary constriction is called as the centromere. Okay, so this is another constriction is also present here, and this is called as the secondary constriction, or it is can called be the satellite. Okay, the first constriction, because it is very much it is inward towards the center, it is called the primary constriction and it is called as the centromere, and the secondary constriction it is here present and now it is called as the satellite. So now look at the structure of the chromatin, that means how many types of the chromatins are present, or if we divide the chromatin, it is two types. One is called as the heterochromatin, another is called as the euchromatin. So one is called as the heterochromatin, another is called as the euchromatin. Look at the structure of the chromosome. So here this dark color, this is the dark strand, they are the dark strand, they are the dark strand, and they are called as the heterochromatin. Okay, so now we can say that the heterochromatins are darkly stained, while the euchromatin, this is euchromatin, they are the euchromatin, so they are the euchromatin, or it is, you can say that they are lightly stained structure that is present inside the chromosome. So what is the function of this heterochromatin, what is the euchromatin? So first come to the heterochromatin. So the heterochromatin, here the genes the present inside the heterochromatin, here and the here, the genes present in the heterochromatins are silenced. So what is the silenced DNA? So that means here the gene that is present, or the DNA sequence that is present in the heterochromatin is silenced. So that means it is not transcriptionally active. That means they are, or these genes are not transcripted. That means the transcription never takes place in that gene that is present in the heterochromatin. So that means here that is called as the silence. That means there is no protein is synthesized from this gene present in the heterochromatin, while in the euchromatin they are very active. That means all the protein are synthesized. After transcription, the mRNA is present, and from the mRNA the proteins are present, or the proteins are formed. So that means all the proteins, or the maximum of the proteins that are formed, or the maximum of the gene that are transcripted, that are comes from the genes that are present in the euchromatin. So that means it is called as the actively genes. That means here the always the transcription takes places. So that's why they are called as the silence gene, or inert genes, or the inert activity or occurs in the heterochromatin, while the euchromatin there the active genes are present. So now, because they are the silence gene, for example, this is the nucleus. So the heterochromatin, because it is the silence or the inert structure, so they are always present in the periphery region of the nucleus. So they are present in periphery region of nucleus, while they are present euchromatin, they are present at the center phase. That means at the central part the euchromatin are present, while at the periphery region the heterochromatin are present. So now the heterochromatin, they are present only in the eukaryotes, while the euchromatin that is present in both prokaryote and eukaryote. Okay. So now the heterochromatin, it can be divided into two parts. One is called as the constructive, another is called as the facultative. Facultative heterochromatin or the constructive heterochromatin. So what is the constructive heterochromatin? That means here the genes are always, or the permanently inactive. That means the genes that are present in the heterochromatin, there is permanently inactive. That means there is never transcription occurs in the this constriction heterochromatin. But in the facultative, that means they are always, or maximum case, the maximum time they are present in the inactive condition, but sometime it transcribes. Okay. What is the example of the facultative heterochromatin? One example is the bar body. The bar body is present, and that is the facultative heterochromatin. That means it can transcribe if needed. So this is the main difference between the euchromatin and the heterochromatin structure. Now come to most important portion of the nucleus, and that is called as the nucleolus. So what is the nucleolus? First of all, the nucleolus, it is a singular term. If more than one nucleus is present, then it is called as nucleoli. And the nucleoli, it is the plural term. That means if more than one nucleus is present, then it is called as the nucleoli. Otherwise, it is called as the nucleolus if it is present in the one number. So this is the structure of the cell. Let's suppose this is all over, it is called as the nucleus. Okay. So now inside the nucleus, at the middle portion, there is a portion like this, or there is a structure like this, and this structure is called as the nucleus. Okay. so nucleolus. So nucleolus, it was discovered by Fontana in 1971. So, and he discovered in eel skin. That means eel is a fish. So, in the eel skin, the Fontana discovered the nucleolus. So, now come to the structure of the nucleolus. So, if you ask what is the structure or the exact structure of the nucleolus, then it is a round color, round, dense and darkly stained structure that is present inside the nucleus is called as the nucleolus. So now the nucleolus, look at the structure. The nucleolus, most importantly, it doesn't have any membrane. So that means, most importantly, nucleolus is a non-membrane structure. That means it doesn't have any membrane structure. So in some organisms, the number of the nucleus is 1. That means we can say that there is a nucleus. For example, in our case, in the human case, so the number of the nucleolus is 1. So you can say that it is the 1. But in many organisms, there are many nucleolus, so we can say that they have nucleoli, because it is a plural term. So that means if the number of the nucleolus is more than 1, then it is called as the nucleoli. So now this nucleoli structure or the nucleolus, because it doesn't have any membrane, so now the content of the nucleus, for example, these are the content of the nucleus and they are called as the nucleoplasm. So what is the nucleoplasm? So what is the nucleoplasm? It is just like the cytoplasm. For example, inside the cell, so they are called as the cytoplasm. So like that, inside the nucleus, the cytoplasm is called as the nucleoplasm. So because the nucleolus is a non-membrane structure, that means it doesn't have any membrane, so that the content of the nucleoplasm is continuous with the nucleolus. That means the content can easily go to inside to the nucleus of the nucleoplasm, or the content present in the nucleoplasm, it can transport to the nucleolus freely because it doesn't have any membrane structure. So now, how the nucleolus is formed? So most importantly, how the nucleolus or this structure, how it is formed. So nucleolus is formed by most important, that is called the NOR. NOR means nucleolar organizing region. So that means, so inside the nucleus many chromosomes are present. So around particular chromosomes, so the nucleolus is formed. For example, let's suppose this is a chromosome. So in the chromosome, around the chromosome, a structure is formed, and that is called as the NOR, the nucleolar organizing region, and it is called as the nucleolus. And it is, one is called as the, this is called as the centromere, or it is also called as the primary constriction. Okay. So now around this part, it is called as the secondary constriction. So that means the nucleolar organizing center is simply, you can say that it is the secondary constriction, and the nucleolus is formed around particular chromosomes, not one. Normally 1 to 6, around 1 to 6 chromosome. For example, this is one chromosome, this is another chromosome, this is again around, this is another chromosome, this is another chromosome, another chromosome, this is another chromosome. So around this, always at the secondary constriction. So by combining this secondary constriction of the NOR region, a nucleolus is formed. So if we ask in the human, around 5 chromosomes, the nucleolus are formed. And if you ask the exact what are the 5 chromosomes around which the nucleolus or the nucleolar organizing region is formed, and the answer will be 13, 15, 20, 21 and the 24th chromosome. That means around these 5 chromosomes, the nucleolar organizing region. So 24 means out of 46. So 13, 15, 20, 21, 24 around which this nucleolar organizing region or the nucleolus is formed. Okay. So now come to the what is the exact structure of the what is the parts of what are the parts of the nucleolus. So there are the mainly 4 parts are present. One is called as the granular zone. The second one is called as the fibrillar zone. The third one is called as the amorphous, or simply you can say that the matrix, or you can say that pars morpha, and the fourth one is called as the nucleolar chromatin. Most importantly, it is the nucleolar chromatin, not nuclear chromatin. Nuclear chromatin means the chromosome that is present in the nucleus. So the nucleolar means the nucleoprotein or the nucleic protein or the rRNA, more exactly the rRNA that is present, and that is called as the nucleolar chromatin, not like the DNA that is present in the chromosomes. So that is present in the nucleus, but in the nucleolar or the nucleolus, there the rRNA is present. So first come to the granular zone. Look at this structure. This is the structure of the nucleolus. At the outer chromatin material, they are called as the perinucleolar material. That means the chromatin material that is present outside the periphery region, it is called as the perinucleolar material, and all the materials, nuclear material that is present inside it, it is called as the intranucleolar chromatin. That means they are present inside or the center of the nucleolus. So first structure is called as the granular zone. Look at this structure. This is the granular zone. So always the granular zone, it is present in the periphery. It is present in the periphery region. So what is the granular part, or what structure that contributes to the granular structure or the granular zone? Mostly it is ribonucleic protein. So what is the ribonucleic protein? Simply you can say that the rRNA and the associated protein is called as the ribonucleic protein. So at the outer region or the periphery region, the granular portion is present, and it contributes, or it contains the ribonucleic protein that contains rRNA and its associated protein, and it is called as the ribonucleic protein. The second one is called as the fibrillar zone. Look at this structure. This is the fibrillar zone, and as the name suggests, the fibril or the fibrillar, that means the fibril like structure that is present inside the nucleolus, and they are present inside or the center part, which is called as the granular portion. So now it contains rRNA plus proteins, and that is the non-histone protein. So in the NEET exam, sometimes it will ask that in which part of the nucleolus the proteins are present, or the ribonucleic protein. If the ribonucleic protein is not mentioned, it is asked that in which part of the nucleolus the proteins are present, the answer will be the granular zone. And if it will ask that in which part the rRNA is present, the answer will be fibrillar zone. Okay. The third one is the amorphous or the matrix or the pars morpha. So look at the structure like the cytoplasm, like the structure that is called as the middle part, or simply it is called as the matrix, or the inside or the center, at the exact center, it is called as the amorphous or the matrix of the pars morpha. And the fourth one is the nucleolar chromatin. So the nucleolar chromatin means here the rRNA or tRNA. So these rRNA, tRNA are mainly present in the nucleolar chromatin along with some DNA structure. Okay. So now what is the function of nucleolus? So if nucleolus is present, so what is the main function of the nucleolus? The main function of the nucleolus means it provides machinery for the protein synthesis. Machinery for protein synthesis. How? So that means if the protein means the transcription is required, transcription, and then translation is also required. So during the transcription and the translation, a rRNA or the ribosome is required, and this rRNA and the ribosome are actually formed in the nucleolus region. So that's why it helps in providing the machinery for the protein synthesis. Which machinery? That is the rRNA and the ribosome. The main second answer is that it synthesizes simply the rRNA and ribosome. So in the NEET exam, it is always asked that what is the site of rRNA in the ribosome. The answer will be the nucleolus. If in the option it will be as that nucleus, nucleolus, both, the answer will be nucleolus. But because exactly inside the nucleolus the rRNA and the ribosomes are formed. So it is the site of the rRNA in the ribosome synthesis. So third one is it also stores the ribosomal protein. Ribosomal proteins. Okay. So these are the main function of the nucleolus. So if you like the video, please like and subscribe. Next come to the nucleoplasm. So nucleoplasm. So what is the nucleoplasm? Simply the cytoplasm like structure that is present inside the nucleus is called as the nucleoplasm. Look at the structure. This is the nucleus, and inside it the cytoplasm like structure or the watery like structure is present, and this water like structure is present, and that is called as the nucleoplasm. So the nucleoplasm, it is also called as the nuclear sap. Nuclear sap, because it is the sap like structure that is present inside the nucleus. That's why it is also called as the nuclear sap, or it is also called as the karyoplasm. So karyo means nucleus and the plasm means like the cytoplasm like structure, and because the cytoplasm like structure that is present inside the nucleus, that's why it is also called as the karyoplasm. So the nucleus, the nucleoplasm, it is simply the matrix structure. That is, matrix means the inside structure. So that's why it is simply also called as the nuclear matrix. That means the structure that is present inside the nucleus. So the nucleoplasm contains chromatin and nucleolus. So this is the nucleolus and the chromatin material that is present inside it, that is called as the chromatin, and that is the main difference between the cytoplasm and the nucleoplasm. So that means what is the cytoplasm and what is the nucleoplasm, or what are the difference between them. So the cytoplasm and the nucleoplasm, they contains mostly the watery substances, and besides the watery substances, the nucleoplasm contains the chromatin, and this chromatin structure that is present inside the inside the nucleoplasm, and that's why it also helps in the, because of the presence of the chromatin, it also helps in the formation of DNA and the RNA, which is absent in the cytoplasm. So that is the main difference between the cytoplasm and the nucleoplasm, that the chromatin material is present inside the nucleoplasm, and that helps in the formation of the DNA and the RNA, which is absent in the cytoplasm. This is the main difference. Another difference is the cytoplasm is less sticky, while the nucleoplasm is highly sticky or highly gelatinous. What is the gelatinous or the highly sticky? That means it is present in more concentrated manner in comparison to the cytoplasm. So they are the two difference between the cytoplasm and the nucleoplasm. One is the presence of the chromatin in the nucleoplasm, another is it is the highly sticky or the highly concentrated structure in comparison to the cytoplasm. So they are the main difference between the cytoplasm and the nucleoplasm. So besides the nucleoplasm or the DNA, also the RNA are present inside the nucleoplasm, and it contains many hydrolytic enzymes. That means the nucleoplasm, it also contains many hydrolytic enzymes, So hydrolytic enzymes means the main difference or the main function of the hydrolytic enzymes means they cleave the biomolecules. For example, the peptidase. The peptidase means it cleaves the protein or the peptide bond. That's why it is called the peptidase enzyme. The peptidase enzyme, the function is to cleave the protein that is present inside the nucleus. Another is the ribonuclease. Nuclease means to cleave the nuclear bond or the nucleus, that means DNA RNA bond, that means to cleave the phosphodiester bond, that means to cleave the DNA and the RNA. The hydrolytic enzymes are also present, and the main important enzymes are also present, that is called as the DNA polymerase. So DNA polymerase, its main function is to, that is the DNA replication. So the DNA replication enzyme, DNA polymerase, is also present inside the nucleoplasm, that helps in the replication of the DNA. So this is the main important function of the nucleus, and another function or the other function are also present. For example, many metabolic activities are also takes place in the nucleoplasm, and again most importantly it also helps in the maintenance of nuclear shape. That means it also helps in the maintenance of the nuclear shape. That means the nucleus shape is also maintained by the nucleoplasm. So this is the main function and the main structure of the nucleoplasm. So if you like the video, please like and subscribe my video. Thank you.